Okay, your slides are projected, and if you would just let us know when you would like them advanced as you give us your talk, we appreciate it, and we're sorry you can't be with us here in North Carolina. Go ahead.
Oh, me too. Me too.
Okay, feel free to start whenever you want.
Okay, well, thank you very much. It's not really a pleasure to be here in my hotel room talking to you all over the phone. I really wish I could be there. I really appreciate the invitation, and I had intended to come and show you some of this work we've been working on in the last 5 years in Europe, in which we've been studying the effects of endocrine disrupting chemicals during development and their role on weight gain and obesity later in life, using multiple approaches: in vitro mechanistic approaches, toxicological approaches, and epidemiology. So we could go to the next slide. I'll be presenting mainly the results of this European project called Obelix, and Obelix was a famous French cartoon character. He's pictured here on the right. He was a very large, strong man, and he was really symbolic for this project, and I'll show you in a moment in my next slide why. So in this project, we were working with 7 institutes in 5 countries in Europe, and the project lasted for 4.5 years and finished at the end of 2013, and we're still busy getting all the papers published, and I'm working on a large integrated review paper that will be submitted soon. So if we go to the next slide, the hypothesis we were setting in this European project was to look at if perinatal exposure, so developmental exposure to EDCs, plays a role in the development of obesity later in life. And if Erin would just click on the animation, you should be seeing Obelix as a young boy, and in this cartoon, Obelix falls into a cauldron of magic potion, which gave him his strength and huge size when he was an adult. So we're wondering, of course, what was in that magic cauldron, magic potion, and was that perhaps endocrine disrupting chemicals? So in this project, we studied various mechanisms that could underlie the exposure effect continuum, and that includes, so if Erin would hit the clicks a few times, changes in adipocyte differentiation, do EDCs during development change birth weight? That could change long-term growth trajectories. Is early growth and body mass index changed in children related to exposure during development? And if you click one more time on the animation, are there changes in hormone and lipid metabolism involved that could underlie these effects? And we were interested in epigenetic effects and focused on, we did some preliminary studies on looking at altered DNA methylation. So one more click there, and you'll see our underlying mechanism there. So if we go to the next slide, and I have to go to it myself, you'll see our approach. This is quite a straightforward approach in which epidemiologists got together with toxicologists, and we really did our best to try to learn from each other and feed into each other's work. The epidemiology consisted of 4 mother-child cohorts from Belgium, the Netherlands, Norway, and Slovakia. And the toxicology involved animal studies and in vitro mechanistic studies. And we used the data coming out of these studies to perform risk assessment to try to understand if current levels of EDCs in our children may play a role in obesity or metabolic disruption. So if we go to the next slide, and if Erin would click a few times, I should have taken out the animation, sorry about that. Just give it a few clicks. You'll see the various compounds we studied in this project. So you'll see with the blue box, we studied these 4 models, these 4 classes of compounds in epidemiological and animal studies. In the yellow box, you see that we performed epi studies also on brominated flame retardants and organochlorine pesticides, but we weren't able to do animal studies with these compounds. And on the right, we did an additional study with bisphenol A only in the animals. And one more click will show you that we looked at in vitro studies in all of these various compounds. So we studied mechanisms looking at a wide range of different chemicals. And we selected these compounds because we were very interested in different types of endocrine disrupting chemicals. It was kind of a fact-finding mission, looking at different endocrine mechanisms that they could disrupt and if this could perhaps be linked to the potential obesogenic effects of these compounds. So if we go to the next slide, you'll see that one of the important studies in Obelix was to get a very detailed idea of exposure in these populations. So what we did was we had various cohorts, which you see in the columns. 2 cohorts from Belgium, 1 from Norway, 1 from Slovakia, and a new cohort in the Netherlands. And you can see how old the cohorts were. Some of them were started back in 2003 or 2004. So the children were up to 6 to 7 years of age at follow-up. And the numbers in the table here indicate how many samples we had of cord blood or of mother's milk from these different cohorts. And the pink box is indicating where we already had analysis available of the various endocrine disrupting compounds, and the green box is showing where we did additional analysis and where we set up some new methods for measuring, for example, phthalate metabolites in cord blood and milk. So if we go to, I think, actually my screen has probably frozen up here. Erin, are you on will you go onto a slide with Europe, various countries in Europe? I can't see where you are actually. Yeah.
She has the map on for you.
Okay, thank you. Because sometimes my internet connection can't show me where you are. So the map of Europe is up, and this is just to show you that we got together with other European birth cohorts. And we got together with 14 different cohorts throughout Europe in order to do one of our first studies, which was a really detailed exposure assessment of PCB and DDE levels in cord blood in these children. And we looked at birth weight. So in the next slide gives you an idea of the range of PCB 153 levels in cord serum. These are up to 8,000 children throughout Europe in various cohorts. And you can see the wide range here from quite low exposure in, for example, the Norwegian and Belgian cohorts. These are given with a red star. And the Slovak cohorts and the Faroe Island cohorts are notoriously high in their POP levels. If you go on to the next slide, it just indicates that with this study we did some associative studies looking at PCB and DDE levels in cord blood and birth weight, and we found significant decreased birth weight related to PCB exposure in these 8,000 children. So this was one of the largest studies of its kind, indicating an association between PCBs and lower birth weight. So we were really curious to know if this lower birth weight would relate to changes in growth and weight in these kids as you follow them up throughout childhood. So if we go on to the next slide, you'll see a design of our human studies. Maybe back one, sorry. Back one slide to the human studies. Yeah, thanks. So the human studies looked at both prenatal exposure to PCBs and DDEs and postnatal exposure, and I'll talk about that in a moment, and also looked at perinatal exposure to dioxin-like chemicals and PFOA. And we followed these children up to 2 years and up to 7 years and looked at growth and body mass index and some metabolic hormones like leptin and adiponectin and insulin. So in the next slide, the animal studies design, you'll see that we designed our animal studies in a similar way, as well as we could, in that we exposed for a 9-week period during gestation and 3 weeks during lactation, so a total of 9 weeks, to 5 different compounds in the diet. So we spiked the diet. We used 8 concentrations, 8 doses that were below the no observed effect level for developmental toxicity. So these were non-toxic low doses. We stopped exposure when the animals were weaned at 3 weeks, and we allowed the animals to grow up to adulthood, up to a year of age. And at the end of the follow-up period, we also gave some of the animals a high-fat diet challenge to see whether the combination of a high-fat diet and this early programming or this early developmental exposure could it play a role in changes in body weight. And you'll see there in the box on the right all the different endpoints we measured in our animal studies. And you can see the mouse strain we used, which was a hybrid mouse strain that we used for our epigenetic studies that would help us to distinguish epigenetic changes in the maternal or the paternal line. So if we go to the next slide, what I'll show you now are just a few results from looking at human studies and then comparing them to the animal studies. For 3 of the compounds we studied many more, but these are just highlights, and they're not all published yet. So they are still will be subjected to intensive review, but some of them have recently been published. So now we're looking at dioxin-like compounds. So as I mentioned earlier, we measured total equivalence, TCDD equivalence in cord blood and in mother's milk. And in the human studies, the models that were used that were corrected for a number of confounding factors, they indicated that there was a significant increase in growth in early life related to perinatal exposure to dioxin-like compounds. So this was related to a child being about 350 grams heavier at 2 years of age, related to this dioxin-like exposure during development. When the children were followed up later in life, up to 7 years, a positive association was found only in girls and not in boys. And in a separate study with a smaller cohort, serum adiponectin and insulin and leptin were measured, and a significant negative association was found with serum adiponectin in both boys and girls in this smaller cohort. So serum adiponectin being, of course, a really important hormone involved in regulating glucose levels and fatty acid breakdown. So we'll go on to the next slide when we compare to what we found in the animal studies with dioxin-like compounds. What you see here are 2 examples of growth curves. I took away the error bars in order just to be able to see the average of 8 animals from 8 different litters. And on the left you see the male offspring, on the right the female offspring. So take into account these were animals that were exposed during development only and early lactational period. The black circles indicate the control group, and the red triangles indicate a dioxin-exposed animals during development. And we see that in particular in the females, the body weight, so these are body weight growth curves throughout life up to 50 weeks of age. Body weight increased significantly in females, and particularly after the challenge with high-fat diet in adulthood. And we found a significant dose-related response here with a benchmark dose. So this is similar to a lowest observed effect level of 453 picograms per kilogram per day during development. This led to a significant increase in fat pad weight in these females. So not only did they get heavier, they became more adipose, so that they developed more adipose tissue, more fat tissue. So in the next slide, just briefly showing the results for PFOA. And PFOA, we found a significant increase in growth in children looking at their growth trajectory up to 24 months. but this did not continue up to 7 years when the children were followed up to seven years. And then if we go to the animal experiments, in the next slide, you'll see that actually here's an example of growth curves for a dose level that's below the no observed adverse effect level for developmental toxicity. You'll see that in these adult animals, in the females on the right, we actually found body weight loss, not weight gain, but weight loss. And this was exacerbated, or it wasn't reversed by the high-fat diet given at the end of life. And this was a significant effect on reduced fat pad weight at a dose of 65 micrograms per kilogram per day, which again is lower than the level that's been used to set the reference dose of PFOA. So here we have examples of chemicals, dioxin showing an effect of an obesogenic effect in females, but PFOA showing actually a weight loss in females. If we go on to the next slide, I'd like to point out here that in our exposure studies with PCB 153, we were able to, because we had such good measures of PCB 153 throughout early development in one of the cohorts, we were able to develop a pharmacokinetic model to model exposure concentrations prenatally before birth and postnatally after birth, making this model depending on total breastfeeding duration and other factors such as the child's weight and maternal age, as you can read here. And this was we published this model, we validated it, and it allowed us to tease out effects of exposure prenatal and postnatal in order to see if that would make a difference, as most of the studies that are out there now are looking at cord blood levels of these compounds, and really not paying significant attention to exposures in the first year of life, in particular through breastfeeding. So if we go into the next slide, you'll see the results of the PCB 153, and I'll only show the postnatal exposure here because we found no association between prenatal exposure to PCB 153 and growth and BMI in children. And recall that I mentioned earlier that we did find effects on birth weight. However, if we look at postnatal exposure and growth in these children, we actually saw that effect on birth weight, that decreased birth weight, continued, and a significant decrease in growth was found up to 2 years of age. Later on, when the children were followed up at 7 years, and we're looking at a fairly large sample size here of over 2,000 kids, there was no association between postnatal exposure and BMI. So it seems at this point that this early effect on growth may not have related to a long-term change in weight. However, in one cohort where we had leptin, adiponectin, and insulin measures at 7 years, leptin was significantly decreased in children from this cohort, indicating that there were changes in important metabolic hormones that could be related to a postnatal exposure to PCB. Going to the next slide, looking at the animal studies, we found, let me see, can you click a few times, Erin, please, so I can get those animations up? We saw no effect actually on body weight or adiposity. Oh, can you go back one? Thank you. No effects on body weight or adiposity of PCB exposure during development. But we did find that there were changes in glucose metabolism in males and in glucagon levels in females at the end of the follow-up period. So this is weeks after the exposure had taken place. So that was a summing up of some of the main results of our epi and animal studies. I'll come back to DDE in a moment. If we go into the next slide, I'll just briefly introduce some of the mechanistic studies we did in Obelix in this project, done by a number of PhD students in the project. There were about 6 PhDs in the project and a number of postdocs. So we've had the pleasure of training a number of young people in this project as well. So if we go on to the next slide, this gives you an idea of one of the major in vitro models we used in Obelix, and that is the 3T3L1 mouse pre-adipocyte cell line. And this cell line is very useful and widely used. It's a pre-adipocyte cell line that you can allow to differentiate by adding a cocktail of hormones to the medium, and this will allow the cells to differentiate into mature cells, and we can quantify the number and the size of these fat cells by dyeing the cells with a fluorescent lipid stain and using some advanced microscopy to quantify these number of fat cells that have differentiated. We're also interested if the exposure to EDCs, to endocrine disruptors, would be accompanied by changes, by epigenetic changes like changes in DNA methylation. So in the next slide, you will see that we studied a panel of endocrine disrupting compounds, and a number of them, including the well-known tributyltin that Professor Bloomberg will talk about this afternoon, induces fat cell differentiation in these cells. But we also found that BDE47 did that as well, and we were interested to see that this brominated flame retardant is also able to induce fat cell differentiation. Some of the chemicals we tested, so like TCDD, actually inhibited fat cell differentiation in this model. If we go into the next cell line, we just showing here some limited dose response relationships between, for example, BDE47 and fat cell differentiation, showing that indeed some of these compounds, like TBT, BPA, BDE47, they induce fat cell differentiation. And if we go to the next slide, you can see that if we measure global DNA methylation in these cells, so basically all of the methylated sites on the genome of these cells, so we collect DNA and measure methylated cytosines in this DNA, we see that global methylation, so there's a general change in methylation being either decreased in the case of TBT or increased in the case of bisphenol A. So this gave us already some indications that this change in adipocyte, the stimulation of adipocyte differentiation is accompanied by changes in DNA methylation. And we were interested to know what genes were involved here. And we measured, we go to the next slide, we measured gene expression of a number of important genes involved in adipogenesis. For example, this gene, the PPAR gamma 2, one of the so-called master regulator of fat cell differentiation, we found that gene expression was highly induced by brominated diphenyl ether 47, by BDE47. And when we looked at methylation of the promoter sequence of the PPAR gamma gene, we found a decrease in methylation. So we were able to show that these effects of BDE47 on fat cell differentiation was accompanied by increased expression of the PPAR gamma gene and a demethylation of the promoter of this gene. So we provided some in vitro evidence of these chemicals affecting epigenetic gene regulation. In the next slide, we did some preliminary work in human cells. So here you see global DNA methylation in cord blood, peripheral blood mononuclear cells. So DNA was collected from cord blood cells, and we measured global methylation in these cells. And what we could find was a modest but significant relationship between DDE levels and hypermethylation of DNA collected from these white blood cells from the cord blood. And interestingly, if Aaron clicks on the animation, I think it should work. You'll see, yeah, thank you. You'll see that, interestingly, DDE is one of the most convincing compounds showing relations between between prenatal exposure and effects on growth and BMI. And in our cohort study, in a recently accepted paper by ESAT, we're finding that prenatal exposure to DDE correlates with early childhood growth, as well as the studies shown by Dania Valvi, who you heard before the break, and her co-workers at CREAL in Barcelona. And we've shown here that DDE may be causing some effects on epigenetic gene regulation. And what we really would wish we could do, but we ran out of time and money, was to look at what genes were being differentially methylated in these populations, and could we come up with some biomarkers. So I'll go to the next slide, please. And this slide indicates that we also did some transcriptomic analysis, and we were looking at some mechanistic analysis of how levels of the compounds we were studying related to different gene expressions. And this work is ongoing still, but it's quite promising in that, for example, DDE is indicating some effects on the glucocorticoid receptor signaling pathway. And this may give us some more candidates to investigate when we're trying to understand how these chemicals could affect growth and body weight in children. So if you go to the next slide, please, I'll just conclude by saying that the Obelix project was able to show or provided evidence, and if you'll click one more time, Erin, provide some evidence that chemicals do indeed play a role in obesity. We certainly saw that these compounds affected growth and metabolic pathways. Not all of them led to heavier phenotypes. Some of them did, but some of them led to showed a leaner phenotype as well. If you click one more time, Erin, you'll see those conclusions coming up. Yeah, and for example, DDE and dioxin-like compounds showing evidence both in animal studies and in epi studies for heavier phenotype, and PCB153 showing evidence for effects on actually inhibited growth in children. We go to the next slide, please. What we're working on, to summarize again, we do believe that the current levels of EDCs may pose a risk for metabolic disruption. We saw very clearly that there is gender specificity in our population, so it really matters if you look at girls or boys. And we saw differences between pre- and postnatal exposure that would require more attention, I think, in the future. And our mechanistic work generated new hypotheses. So I think a number of speakers have already mentioned, for example, we need to look at effects of mixtures of chemicals, and we need to look at long-term consequences of these early changes in growth. We know from studies that early childhood obesity is a predictor of long-term obesity. So for some of these relations that we found, we are concerned, and we'd really like to follow up these kids much longer. So we go to the next slide, please. And I'd just like to show you just really briefly some new work coming out of my lab. We are following up these cohorts within another European project that focuses on neurodevelopment. And in my own lab, we've been working for the last 3-4 years on an alternative model of obesity, the zebrafish model. Zebrafish are a really interesting, useful, small vertebrate model. We can perform multi-generational epigenetic studies in them, which we're doing, which are ongoing at the moment. And we've also been looking at effects of disrupting circadian rhythms and how that can affect fat cell differentiation. So you see in the figure at the bottom here, this is a little fish, only 15 days old. The fish is still quite transparent, and if you stain the fish, the living fish, with a lipid stain, in this case it's called lipid green, those little white balls that the arrow is pointing to, those are fat cells that are forming between 12 and 15 days in the fish, and you can visualize them and quantify them in a living organism. That makes this model really useful, I think. And if you go to the next slide, what we've been doing recently is working with a transgenic zebrafish line that actually you can monitor the activity of the clock genes. So clock genes are really important in circadian rhythms. And this transgenic line that we're using, you can see the graph on the right there. You can monitor the luciferase reporter gene activity, which is under control of a promoter sequence for the clock and BMA1 genes. These are important clock genes. And you can see this daily pattern of high peaks of activity and low peaks of activity. This is a normal pattern of clock gene activity throughout the 24-hour period in a fish, but also in a human. And if you go to the next slide, you'll see that we started some work to understand if obesogenic chemicals can disrupt circadian cycles as well. It's a bit of a busy slide, and it's very preliminary work. But what you see on the left are pictures taken of fish, and all you see are some red fluorescence, and that red fluorescence is lipid staining in a 15-day-old fish. And you can already see just from the pictures that the fish exposed to TBT and benzophenone 3 in the bottom right-hand panel there. That's a chemical we heard about from our previous speaker, a phenolic UV filter. They really enhance lipids accumulation in these fish if you give them, expose them to low concentrations during development. And if you look on the right, on that really busy slide there, it's just showing you that the circadian cycling, so the clock gene expression of these fish, is really screwed up if you expose them to these chemicals. So the dark blue squares at the bottom are actually benzophenone 3 exposed fish, and they show absolutely no circadian rhythm anymore when they're exposed to benzophenone 3. So we're really wondering, what's the chicken and what's the egg? Are these chemicals affecting circadian rhythm, and that's affecting metabolism, or the other way around? Is metabolism affected by obesogenic chemicals that disrupt circadian cycling? In any case, it's a really fascinating project to try to understand maybe alternative mechanisms by which chemical exposure can affect metabolism. So I'll conclude by that. If you go to the next slide, please, you'll see all the wonderful people who worked on this project. If you click a million times, Erin, you'll see all the names. And I would like to thank all of these people. And I would like to thank you and very much for your attention. And I wish I could be there. Thank you.
Thank you very much, Juliet. We're sorry you couldn't be here, too. While the other panel members are convening in the front, if anybody has any clarifying questions for her, why don't you go to the microphone in the back and introduce yourselves. And then our two speakers from this morning, if you'll come up to the panel table.
So I have a very simple question, and that is, has anyone measured the level of these obesogens in animal chow? Is the control really zero?
Oh, that's a good question. Can you hear me?
Yes, we can.
Yeah, right?
Mm-hmm.
Yes, that's a very good question. Yes, of course. Well, in our animal studies, we do measure the levels in the chow and the levels in the serum as well. And you do find some background level, of course, certainly in, for example, in our phthalate exposures, which I didn't show you. You will find low background levels already in our chow, and we do our best to avoid background contamination, using appropriate containers and appropriate avoiding plastics, etc. But you will never completely avoid it.